We've all felt that, familiar misery of a flu or a persistent cough. But what we rarely consider is the incredibly complex, high-stakes biological warfare happening inside us.
It really is warfare.
Today, we're embarking on a crucial deep dive into the general principles of microbial pathogenesis.
That's our mission. And we aren't here simply to list off diseases. We're providing you with a thorough, structured understanding of the rules of engagement in this biological battlefield.
Right.
We're going to trace the microbe's journey, how it breaches our defenses, how it evades the immune system, and critically, how disease results not just from the microbe's action, but often from the collateral damage caused by our own necessary response.
And understanding these rules is globally relevant. I mean, infectious diseases are tragically still a leading cause of death worldwide.
Absolutely. Especially in low-income countries where issues like diarrheal diseases and acute respiratory infections take just a monumental toll.
So for you, the learner, the goal today is to move past just the symptoms and really understand the mechanisms, the virulence factors, the invasion pathways, and the specific host responses that generate those effects we all feel.
It's truly the ultimate chess match of life. By the end of this deep dive, you'll understand the specific evolutionary advantages that allow a pathogen to survive the human body's sophisticated defense systems.
And how, in many cases, its success relies on manipulating our very own immune cells.
Exactly, turning our strengths against us.
So to begin the story of pathogenesis, we have to start at the border. Our bodies are essentially walled fortresses, complete with multiple layers of defense. Section 1 is all about how the microbial invaders find the weak points and bypass the guardians.
That's the perfect way to frame it. Think of the human body as having, say, 4 major entrances, each guarded by a specific epithelial barrier. These barriers are the physical and chemical first lines of defense, and for a pathogen to succeed, it must either overwhelm them or find a back door.
Let's start with the biggest one, the skin.
Right. The skin. It's our largest organ, and fundamentally, it's designed to repel. Its outer layer is composed of these tough, keratinized cells. It's a structural shield.
It's literally armor.
It is. But beyond that physical structure, it's also chemically hostile. It maintains a low pH and secretes fatty acids, which actively inhibit the proliferation of many potential invaders.
So if the walls are that robust, how does the fortress fall? It sounds like a full frontal assault is usually futile.
That's the key insight. Successful microbial entry through the skin is rarely due to a microbe simply dissolving its way in. It's predominantly a failure of mechanical integrity.
Meaning a cut, a scrape.
Exactly. We're talking about trauma, burns, surgical incisions, or needle punctures, any breach that physically bypasses that tough keratinized layer.
And we shouldn't forget the tiny but mighty mechanical failures caused by nature itself, the insect vectors.
Oh, absolutely. Mosquitoes, ticks, fleas, they are sophisticated biological syringes. They pierce the barrier and inject the pathogen directly into the bloodstream or the underlying tissue.
Giving the pathogen an immediate and complete bypass of all the skin's defenses.
Total bypass. Now, there are exceptions. We do have fungi, the dermatophytes, which cause superficial infections like athlete's foot. They literally colonize the keratinized surface itself, setting up shop where the chemical defenses just aren't strong enough to halt their growth.
Okay, moving inward, we face the next major challenge, the gastrointestinal, or GI, tract. This sounds like a true gauntlet of chemical defenses.
It is an incredibly harsh environment. The stomach, of course, features extreme acidity, which is immediately bactericidal for most organisms you might swallow.
Kills them on contact.
Pretty much. Then you move into the small intestine, which utilizes a whole cocktail of digestive enzymes, bile salts, and various mucosal immune defenses.
I remember you calling the stomach acid the VIP defense. How critical is that acidity, really?
Oh, it is crucial. It acts as a continuous sterilization process. But what's fascinating is how we know this. If a host's stomach acid is neutralized, either therapeutically by medication like an antacid,
Or if you just ingest a massive dose of the microbe.
exactly. If the microbe is ingested in such an overwhelming dose, it can actually buffer the acid, and vulnerability skyrockets. A huge dose of Vibrio cholerae, for example, essentially overwhelms that primary gate, neutralizes the acid, and allows the survivors to proceed further down the tract.
And beyond the chemical defenses, the GI tract has active physical defenses, doesn't it? Peristalsis, for instance.
Yes, the constant physical churning of peristalsis acts as a flushing mechanism. It prevents microbes from staying in one place long enough to establish an infection.
Just keeps things moving.
Right. And perhaps the most complex defense is the normal gut microbiota, this vast, complex ecosystem of trillions of commensal bacteria. They occupy receptor sites and actively produce antimicrobial compounds. It's a powerful colonization resistance against new invaders.
It's like the good guys have already taken all the parking spots.
That's a great way to put it. So for successful invaders like Vibrio cholerae or Shigella spp to triumph, they need highly specialized tactics.
How do they do it?
Well, Vibrio cholerae focuses on rapid colonization and attachment to the mucosal surface, surviving the stomach through that high dosage we mentioned. Shigella, on the other hand, is a master of invasion. It successfully breaches the epithelial layer and invades the actual cells of the colon, allowing it to multiply out of reach of all those defenses in the lumen.
Okay, next up, the air we breathe, the respiratory tract. We are constantly inhaling potential pathogens. What keeps that system clean?
The respiratory system relies on two main highly efficient systems. The upper tract is dominated by something called mucociliary clearance.
The mucociliary escalator.
The escalator, exactly. It's a conveyor belt of ciliated epithelial cells that sweep microbes trapped in mucus up and out of the lung. But if a microbe makes it deep into the alveoli, it faces the resident alveolar macrophages.
The frontline police force.
The police force deep in the lung tissue, yeah. They're designed to engulf and kill any particulate matter, including microbes.
It sounds comprehensive.
Yeah.
So how does something like influenza or M. tuberculosis manage to win?
They launch a counterattack targeting the mechanism itself. Influenza viruses actively target and destroy the ciliated cells.
So they break the escalator.
They break the escalator, effectively shutting down the conveyor belt and leaving the respiratory surface vulnerable to both the virus and, importantly, secondary bacterial infections.
And Mycobacterium tuberculosis.
It's even sneakier. It gets engulfed by the alveolar macrophage, just like it's supposed to, but it has evolved mechanisms to resist the killing. It actually multiplies within the very cell designed to destroy it. A powerful evasion tactic we'll definitely circle back to.
Wow. Okay, finally, let's quickly address the urogenital tract. This system relies less on complex barriers and more on, well, simple mechanics.
That's accurate. The primary protection here is the mechanical flushing mechanism provided by regular urination, which prevents attachment. In the female tract, the indigenous vaginal microbiota maintains an acidic environment, which is crucial for inhibiting the growth of most invading pathogens.
And the failures here often seem linked to disruption or trauma.
Precisely. Infections often take hold when the flushing is impeded, maybe by an obstruction, or when local trauma provides an entry point. A really common example is the use of broad-spectrum antibiotics.
Which wipes out the good bacteria along with the bad.
Right, and that can wipe out the protective, normal microbiota, allowing opportunistic organisms like Candida albicans to overgrow and cause localized infections like candidiasis or a yeast infection.
Okay, so the pathogen has breached the barrier. It's in. But it's not done. It has two simultaneous objectives.
Yeah.
First, ensuring its continuation by finding a way to get out of this host and into the next.
And second, spreading systemically within the current host to establish a more serious disease. Let's look at those movement strategies.
The microbe's survival depends entirely on efficient spread from host to host.
It does, and the means of release are directly dictated by the microbe's preferred habitat and how well it can survive outside the body.
We see everything from the simple to the highly organized.
Indeed. Simple skin shedding and contact are routes. Then you have expulsion, coughing, sneezing, salivary transfer. These transmit highly contagious respiratory pathogens like measles and varicella zoster virus. The explosive force of a sneeze is a highly efficient dissemination mechanism.
And the direct routes, obviously, like sexual contact, which transmits pathogens that are fragile outside the body, such as Treponema pallidum, the agent of syphilis.
And we can't overlook the fecal-oral route, which is crucial for pathogens that can survive the harsh GI tract and water systems like polio and certain strains of E. coli. The microbe needs to survive its passage through the GI system of one host and then be ingested by the next.
And finally, vector transmission provides a clean, immediate path.
A very clean path. Arthropod vectors, like mosquitoes, transmit malaria and yellow fever, bypassing the epithelial barriers entirely. Or direct bites from animals can transmit viruses like rabies.
So that covers host to host. Now, for the systemic movement within the current host, How does a localized infection become a full-blown systemic disease affecting the brain or the heart?
Most pathogens initially multiply at the site of entry, but to cause serious, deep-seated disease, they have to utilize the host's circulatory systems, the lymphatics, and the blood.
So it's often a sequential process. They don't just jump straight into the blood.
Not usually, no. A pathogen frequently spreads first to the local lymph nodes, draining the site of infection. It multiplies there, perhaps even resisting the immune cells, and then uses the lymphatic drainage system to gain access to the main circulatory system, the bloodstream.
And that results in viremia for viruses or bacteremia for bacteria.
Correct. And that systemic spread, that bloodborne dissemination, is what escalates the danger. Once they are in the blood, the entire body is suddenly vulnerable.
It's a superhighway.
It's a high-speed transportation network, exactly. It allows the pathogen to reach vital organs, the liver, the heart, the spleen, the brain. This is what leads to profound diseases. For instance, in severe bacteremia, bacteria can seed the heart valves, causing endocarditis.
But some viruses bypass the circulation almost entirely, using the nervous system instead.
This is a remarkable adaptation. Nerve spread. Viruses like rabies and varicella zoster have evolved the machinery to exploit peripheral nerves, traveling up the axons to reach the central nervous system directly.
That sounds slow, but very stealthy.
It's incredibly efficient in its own way and allows rabies, for example, to reach the brain completely undetected by the immune cells circulating in the blood.
That transition point from simple local infection to systemic threat is where the real conflict begins. Once the microbe is moving, it immediately encounters the full arsenal of the adaptive immune system.
And that leads us directly to the sophisticated countermeasures and the damage they inflict, which brings us to Section 2.
Right. The sheer genius of microbial pathogenesis is not just in breaching the gates, but in their highly evolved strategies to actively fight back against a highly organized immune defense system.
This section is all about those offensive weapons and the resulting collateral damage. The crucial takeaway here is that microbes have evolved specialized tools, we call them virulence factors, to survive this encounter. It's an evolutionary arms race.
And one of the most dynamic ways a pathogen survives the onslaught is through antigenic variation.
This is where the pathogen changes its surface structure, its uniform, to avoid detection by antibodies the host has already produced.
It's like a spy constantly changing disguises.
Perfect analogy. If the host mounts an effective antibody response against surface protein A, the pathogen rapidly switches to express surface protein B, rendering all those existing antibodies completely useless.
We see this with HIV, right? It's high mutation rate.
Exactly. HIV is constantly generating new antigenic variants, and we also see it in the influenza virus, which utilizes genetic reassortment or antigenic shift. This allows entirely new pandemic strains to emerge because the entire population has no prior immunity to the new coat.
And the mechanism is even more complex in parasites. Tell us about the trypanosomes.
Trypanosoma brucei, which causes African sleeping sickness. It is the textbook example of this mechanism taken to an extreme. It doesn't rely on random mutation. It uses a process called gene conversion to sequentially switch the expression of its variant surface glycoprotein, or VSG.
So it has a whole wardrobe of coats ready to go.
A massive wardrobe. The host produces a huge wave of antibodies to the current VSG, clearing most of the parasites. But a few individuals in the population have already switched their gene expression. They evade detection and lead to the characteristic cyclical waves of fever and disease that define the illness.
Beyond changing identity, some microbes employ simple blocking and hiding techniques to thwart the immune system's cleanup crew, the phagocytes.
Evading phagocytosis is a fundamental requirement for many successful pathogens. The simplest strategy is a protective coating. Streptococcus pneumonia, for instance, utilizes a large polysaccharide capsule.
It just makes itself too slippery to grab.
That's a great way to think about it. The physical shield inhibits phagocytosis because it makes the bacteria too smooth or bulky for the macrophage to bind to and engulf effectively.
But the truly masterful microbes don't just block the process. They allow themselves to be engulfed, only to survive and multiply inside the very cell designed to kill them.
These are the intracellular survival specialists. Think Listeria monocytogenes and Mycobacterium tuberculosis. When they're phagocytosed, these organisms actively prevent the phagosome, that's the membrane-bound bubble holding the microbe, from fusing with the lysosome, which is the vesicle packed with potent digestive enzymes and toxic oxygen radicals.
So, if the phagosome is the delivery truck and the lysosome is the incinerator, The microbe is stopping the truck from ever docking.
That's a perfect analogy. It prevents the delivery of the killing machinery. Furthermore, Listeria takes it a step further. It produces an enzyme that allows it to escape the phagosome entirely, moving into the host cell's cytoplasm.
And once it's in the cytoplasm, it's totally hidden from things like antibodies.
Completely hidden from humoral immunity. It can multiply safely and even use the host cell's own machinery to move directly into adjacent cells, avoiding the extracellular immune system entirely.
A completely different but equally effective hiding strategy is latency, which is favored by viruses.
Latency is the viral equivalent of going dormant in a hidden bunker. Viruses like the herpes viruses, so HSV, VZV, CMV, EVV, they enter a state where they replicate minimally, or not at all, typically residing silently within neurons or lymphocytes.
And this keeps them hidden from the host's cytotoxic T cells, which are the primary soldiers for clearing active viral infections.
Precisely. Because there's minimal active viral protein being produced, the host cell displays few, if any, viral antigens on its surface. This allows the virus to persist silently for years, sometimes decades, only reactivating when the host's T-cell immunity wanes.
And finally, we have pathogens that don't just hide or change, they actively suppress immunity. They target the core functionality of the host's defenses.
This is direct sabotage. The classic example is HIV, which selectively infects and systematically destroys CD4 plus T helper cells.
The generals of the immune army.
The generals, the central commanders of both humoral and cell-mediated immunity, their destruction leads directly to the collapse of the entire adaptive defense system, resulting in AIDS.
And other viruses use more subtle biochemical sabotage.
They do. Cytomegalovirus, or CMV, produces specific proteins that interfere with antigen presentation pathways. It even generates molecules that mimic human immune regulators, like homologs of the anti-inflammatory cytokine IL-10. It effectively turns down the host's alarm system.
So it's telling the immune system, everything's fine here, nothing to see.
Exactly. And Epstein-Barr virus, during chronic infection, can induce what's called T-cell exhaustion, meaning the specific T-cells generated to fight the virus become functionally depleted, leading to a diminished capacity to clear the persistent infection.
Okay, so the pathogen is inside and it's evading the guardians. How does the actual disease, the tissue destruction, occur? We established that it's often a combination of the microbe's direct weaponry and the host's overzealous response.
That distinction is vital. Let's start with the direct hits, particularly from viruses, direct cytopathic effects.
Viral damage often involves cell death, which is sometimes intentional and sometimes just a byproduct of the virus using the host cell's machinery.
Right. Viral replication can lead to cell lysis, physically bursting the cell membrane to release new viruses, or the virus can trigger the cell's programmed self-destruct mechanism apoptosis by activating death receptors on the cell surface.
And what about those syncytia?
Syncytia. What's also crucial is the ability of some viruses, like herpes viruses, to cause infected cells to fuse with their neighbors. This creates these large, multinucleated giant cells.
And what's the advantage of that?
It allows the virus to spread from cell to cell without ever leaving the protective cell environment and encountering antibodies in the bloodstream. It's a way to spread under the radar.
And we must mention the transformative aspect viruses causing uncontrolled growth or cancer.
Yes. Viruses like human papillomavirus, HPV, and hepatitis B and C can insert their genetic material into the host cell's DNA. This can alter cellular growth and metabolism and drive uncontrolled proliferation. This capacity for transformation is what links these pathogens directly to oncogenesis.
Okay, turning to bacteria. Their most spectacular direct weapon is the toxin. Let's start with the secreted proteins, the exotoxins.
Exotoxins are incredibly potent, specific, and often lethal protein molecules secreted by bacteria. They are purpose-built to interfere with host cell function or kill the cell outright.
The sources categorize the AB toxins as, well, molecular masterpieces of targeted assassination.
I think that's fair. The structure is key. The B subunit is the binding component. It acts like a highly specific key to unlock a receptor on the host cell. The A subunit is the active enzymatic portion, which is then delivered inside to perform the toxic function.
So B is for binding, A is for active.
Exactly. Diphtheria toxin, for example, targets the protein synthesis machinery inside the cell, essentially shutting down the host cell's ability to live. Cholera toxin interferes with electrolyte transport, causing that massive watery diarrhea.
And outside of the AB structure, we have membrane-damaging toxins like the hemolycins produced by Staphylococcus aureus. They just punch holes in cell membranes.
Correct, causing lysis. But if we want to talk about true immunological chaos, we have to spend serious time on superantigens.
Okay.
These are a specialized class of exotoxins produced by certain Staphylococcus and Streptococcus strains.
And this is where the complexity jumps significantly. Superantigens bypass the highly regulated process of normal antigen presentation, leading to a catastrophic overreaction.
They are essentially biological brute force mechanisms. In a normal immune response, only T cells specific to a particular antigen are activated, maybe 1 in 10,000 T cells.
The very specific targeted response.
Very. A superantigen, however, physically links the T cell receptor to the MHC2-IA molecule on the antigen-presenting cell, regardless of the peptide antigen being presented.
This means they trigger massive, non-specific T cell activation.
It's an immunological tidal wave. Instead of activating 0.01% of your T cells, you see anywhere from 5 to 30% of all T cells system-wide activated simultaneously.
To 30%. Wow.
This forced activation results in the uncontrolled, massive release of inflammatory cytokines, TNF, IL-1, interferons, a phenomenon known as the cytokine storm.
And that cytokine storm is the definition of fatal friendly fire.
The systemic consequences are devastating. Massive vasodilation, systemic capillary leakage, coagulation defects, shock, and multi-organ failure. The full spectrum of toxic shock syndrome. The severity of the disease is entirely attributable to the host's inability to control its own inflammatory response.
The second major class of bacterial weapons is endotoxins, specifically lipopolysaccharide, or LPS.
Right, which is found in the outer membrane of all gram-negative bacteria. Unlike exotoxins, which are secreted proteins, LPS is part of the bacterial structure. It's released in high quantities only when the gram-negative bacteria die in the hand.
Which is a crucial difference.
Yes.
So if a patient has a massive gram-negative infection and is given a high dose of antibiotics that causes rapid bacterial death, the initial result could actually be worse, not better.
That is the terrible irony of endotoxin pathogenesis. The rapid bacterial clearance, which should be beneficial, can trigger a dramatic inflammatory spike as huge amounts of LPS are suddenly released into the bloodstream.
What does the LPS do?
The lipid A component of LPS stimulates host cells, primarily through the toll-like receptor 4 pathway. This leads to the rapid release of massive inflammatory cytokines, TNF, IL-1, and IL-6. This cascade results in high fever, profound systemic vasodilation, the collapse of blood pressure septic shock, and widespread microvascular injury. Endotoxin is the primary driver of gram-negative sepsis.
Okay, so the symptoms we feel, fever, pain, swelling, are the visible signs of the immune system's battle plan. But the pattern of inflammation can provide forensic evidence about the type of pathogen involved.
It can. Our sources categorize tissue reactions into 5 distinct patterns. The first and most common for many bacterial infections is suppurative or purulent inflammation.
This is the pus-forming reaction dominated by neutrophils.
Exactly. Suppurative inflammation involves increased blood vessel permeability and the massive influx of neutrophils, our first responders, to the site of infection. The result is the formation of pus, which is essentially a collection of dead neutrophils, dead bacteria, and tissue fluid.
So like in bacterial pneumonia or a classic abscess from S. aureus.
Precisely. The second pattern is far more indicative of a persistent, intractable fight, mononuclear and granulomatous inflammation.
This defines chronic infection.
It does. Instead of acute neutrophil-dominated pus, the battlefield is characterized by monocytes, macrophages, and lymphocytes. This reaction occurs when the host response fails to eliminate an antigen, perhaps because it's hiding inside macrophages or is chemically indigestible.
And the result is the formation of a granuloma, which we see in tuberculosis and syphilis. What is the functional significance of this structure? Why build it?
The granuloma is the body's ultimate containment strategy. Activated macrophages, unable to kill the persistent microbe, aggregate into these dense nodules, often surrounded by lymphocytes and fibroblasts. They're effectively building a physical wall around the threat.
But that wall has a cost.
It has a huge cost. While it limits systemic spread, the chronic inflammation and subsequent necrosis within the wall, that cheesy center, caseous necrosis, lead to extensive tissue destruction and scarring over time. The pathology is the body's attempt to contain the threat.
Moving on, we have reactions dominated by viral effects. Cytopathic cytoproliferative reactions.
These reactions are defined by the microscopic effects viruses have on host cells. We see necrosis, the formation of those multinucleated giant cells, or syncytia we mentioned earlier, or the development of inclusion bodies.
Which are what, exactly?
Visible aggregates of viral proteins or particles inside the host cell nucleus or cytoplasm. Herpes viruses and CMV infections are classic examples where you can see these under a microscope.
And finally, reactions that simply involve wholesale tissue destruction.
Yes, tissue necrosis. Think of the rapid death of muscle tissue seen in gas gangrene caused by Clostridium perfringens. And perhaps most common in chronic viral diseases is chronic inflammation and scarring. Here, the long-term low-grade inflammatory response leads to the deposition of collagen and fibrosis, ultimately causing organ dysfunction.
Like in chronic hepatitis B and C infections leading to liver scarring and cirrhosis.
Exactly. The host's own response destroys the functional architecture of the organ.
Okay, let's take these general principles of entry, evasion, and damage and apply them. This is where the mechanisms become concrete, offering those deep insights into why specific diseases manifest in specific ways. We'll begin with viruses.
Sounds good. Let's start with Zika virus, a flavivirus. It's a relatively mild pathogen in adults, yet a devastating threat congenitally.
Zika gained global attention because of its link to severe birth defects. Its transmission methods, mosquito, sexual, and congenital, highlight its flexibility as an invader.
What really defines Zika's pathogenesis is its cell tropism. While many viruses infect general tissue, Zika is highly neurotropic. It preferentially targets nervous tissue.
But more specifically than that.
Much more specific. It replicates aggressively in neural stem cells within the developing fetal brain.
So if the virus is attacking the very cells responsible for building the brain, the consequence has to be profound.
The result is the catastrophic developmental failure we see in microcephaly, cerebral atrophy, and severe brain developmental issues. And the timing is crucial. Our sources indicate that if the infection occurs during the first trimester of pregnancy, the risk of severe birth defects can be as high as 40% in some populations.
Because that's the period of peak fetal neurogenesis.
Exactly. The aha moment here is understanding that the severity isn't due to a powerful toxin, but the incredible precision of its cellular target.
Next, measles virus, a paramyxovirus. This is a classic example of a respiratory entry leading to systemic dissemination.
Measles is perhaps the most contagious common human virus. It enters via the respiratory route, but its primary replication occurs in the respiratory epithelial cells. Crucially, it then uses white blood cells lymphocytes as a taxi service.
To travel via the bloodstream and disseminate throughout the body.
Yes. And this systemic spread is why the rash is so widespread, affecting the skin and other organs. And let's not forget the characteristic lesions in the mouth.
The Koplik spots.
The Koplik spots, yes. Ulcerated necrotic lesions in the oral mucosa. They're often the earliest diagnostic sign, reflecting that systemic spread. But while the acute disease is harsh, it's the long-term threat that is particularly frightening. Subacute sclerosing panencephalitis, or SSPE.
SSPE is a rare but almost universally fatal complication that manifests years after the initial infection has seemingly cleared.
It's an example of incomplete viral clearance. A defective strain of the measles virus persists, establishing a slow, chronic, smoldering infection within the neurons of the central nervous system. This highlights the concept that even successful clearance of an acute infection doesn't guarantee the complete eradication of the pathogen,
leading to devastating delayed consequences decades later. Our final viral case study is varicella-zoster virus, or VZV, which shows off the latency principle perfectly.
Right. VZV causes chickenpox, a highly contagious systemic infection upon initial exposure. After this acute phase, the virus utilizes the mechanism of latency. It hides out silently in the sensory neurons of the dorsal root ganglia near the spinal cord.
So if the immune system essentially wins the first battle, why does the enemy remain stationed inside the territory?
It's a strategic retreat to a secure bunker. The neurons provide an immunologically privileged site where the virus can persist for decades, largely invisible to T cells. The subsequent eruption of shingles, or herpes zoster, is VZV reactivation.
Typically triggered years later by a decline in the host's T-cell immunity, often due to age, stress, or immunosuppression.
That's right. And the resulting pathology is incredibly specific because of that neuronal latency.
How so?
When VZV reactivates, it travels down the peripheral nerve axons to the patch of skin supplied by that specific nerve, the dermatome. This explains why shingles causes that painful, localized vesicular rash, strictly limited to one side of the body, following the nerve distribution. The localized disease is a direct reflection of its site of latency and its pathway of spread.
Switching gears to bacteria, we look at two invaders that rely heavily on manipulating the host's immune response over years to cause their most devastating damage. tuberculosis and syphilis.
Tuberculosis, caused by Mycobacterium tuberculosis, is the quintessential intracellular pathogen. It relies on evasion tactics to generate a massive chronic inflammatory response that really defines the disease.
The battle begins when the microbe enters the lung and is immediately engulfed by an alveolar macrophage.
But the TB bacterium is prepared. It resists intracellular killing by actively preventing that crucial phagosome-lysosome fusion we discussed earlier. This allows it to multiply unhindered within the macrophage cytoplasm. This survival mechanism is the bedrock of its pathology.
And this triggers the formation of the primary complex, a localized lesion in the lung, and associated lymph nodes known as the Gon complex.
This complex is a product of a robust but failing T-cell immune response. If the host mounts a strong Th1 response, it attempts to contain the proliferating bacteria by forming that dense protective granuloma, that wall of activated macrophages and lymphocytes surrounding a necrotic case-eating core.
And for most people, that wall holds.
For about 90% of immunocompetent individuals, this complex heals or remains latent, walled off, and sequestered.
But the wall is only as strong as the host's immunity.
Exactly. Secondary, or reactivation, TB occurs years later, typically in the upper lobes of the lungs when host immunity wanes. The walled-off bacteria reactivate, leading to progressive tissue destruction, the formation of cavities in the lung, and the high risk of miliary TB, where the bacteria spread widely through the bloodstream.
So the irony is that the body's initial success in walling off the infection creates the conditions for its later, more destructive failure.
That's a perfect summary of it.
Our second example, syphilis, caused by Treponema pallidum, is often called the unseen invader because it's a fastidious spirochete that can't be cultured in a lab. Its pathology is defined by its ability to elicit a slow, multi-stage response over decades.
T. pallidum progresses through 3 distinct clinical stages, each driven by a different intensity of immune engagement. In the late stages, tertiary syphilis, we see the full force of chronic immunopathology.
This is where we encounter the gummas. How do these destructive lesions relate back to the granulomatous inflammation we just discussed with TB?
They are pathologically similar. Gummas are chronic, granulomatous lesions nodules of destructive, necrotizing tissue surrounded by mononuclear inflammatory cells. While they contain very few spirochetes, they represent the massive, chronic host attempt to wall off the organism.
And this leads to devastating damage to bone, skin, and organs.
It does. Unlike the highly infectious primary and secondary lesions, the tertiary gumma is largely a non-infectious, destructive immunological phenomenon.
And the risk extends to the cardiovascular system, making tertiary syphilis a threat to the heart itself.
Yes. The chronic inflammation can localize in the walls of the aorta, causing aortitis. This weakens the vessel wall and often leads to dilation and subsequent aortic valve regurgitation, potentially causing heart failure. This is another classic example of immunopathology. The inflammation, intended to clear the spirochete, slowly but surely destroys the functional tissue of the cardiovascular system.
And before we move on, we have to touch on the devastating consequence of congenital syphilis.
If the infection is transmitted in utero, especially during the mother's secondary stage, the consequences for the fetus are severe. It can cause immediate infantile symptoms or delayed complications that manifest years later.
Like deafness, or those characteristic notched Hutchinson teeth.
Exactly. And the saddle nose deformity. These are physical manifestations of chronic, unresolved inflammation and tissue destruction during development.
We've seen the calculated attacks of viruses and bacteria. Now we move to 2 distinct classes of pathogens defined by unique structures and life cycles: fungi and parasites.
And these often expose the vulnerabilities of the immune system, either through opportunism or through sheer biological complexity. Fungi often pose a threat primarily when the host is compromised, while parasites are masters of biological complexity, relying on multi-stage life cycles that guarantee transmission and long-term persistence, often leveraging vectors.
Let's start with Candida albicans, the most frequent fungal pathogen. It really exemplifies opportunism.
It does, and it transforms dramatically based on the host's status.
Candida is a dimorphic fungus. Tell us why that structural flexibility is a key virulence factor.
Dimorphism means it can switch between growing as a simple yeast cell and forming long, branching filamentous structures called hyphae and pseudohyphae. This morphological switch is crucial.
Why is that?
Because the yeast form is often harmlessly commensal, just living on our surfaces without causing disease. But the hyphal form is what allows the fungus to actively invade tissues.
It causes common superficial infections, thrush, diaper rash, vaginitis. But the real danger emerges in the immunocompromised.
Correct. In hosts with severely impaired immunity HIV, chemotherapy patients, or those on prolonged broad-spectrum antibiotics, Candida can breach epithelial barriers and enter the bloodstream, becoming systemic.
And that leads to serious, deep-seated infections like meningitis or endocarditis.
Right. Its virulence depends on a few factors. Adhesins to stick to surfaces like catheters, the hyphae for active invasion, and secreted proteases to enzymatically degrade host tissue. It is an enemy just waiting for the castle walls to be temporarily unmanned.
Next, Cryptococcus neoformans Gotti, which utilizes a highly effective physical shield.
The defining pathogenic feature of Cryptococcus is its large, thick polysaccharide capsule. This capsule is enormous relative to the yeast cell itself, creating a physical barrier that mechanically resists recognition and engulfment by host phagocytes.
It's an effective cloak of invisibility.
It is against the innate immune system, and this resistance to phagocytosis directly links to its status as a major cause of severe disease.
Right.
Because it is poorly cleared by the initial immune response, it can spread hematogenously, and its propensity to cause meningoencephalitis in immunocompromised patients, particularly those with AIDS, makes it a highly feared pathogen. We know it's found ubiquitously in soil and pigeon droppings, and that capsule is the key to its pathogenic success inside the human body.
Moving to parasites, we jump to organisms with incredibly complex life cycles designed for maximum persistence and transmission, starting with malaria, Plasmodium spp.
Malaria, transmitted by Anopheles mosquitoes, affects over 200 million people annually and remains one of the greatest global health burdens. Its complexity lies in its multi-stage life cycle within the human host.
The journey begins with the mosquito injecting sporozoites, which first head straight to the liver.
That's the exerythrocytic stage. Sporozoites infect liver cells, where they multiply rapidly and asexually. Once mature, they release thousands of merozoites, which transition the infection to the erythrocytic stage by invading red blood cells.
And this is the stage that causes the classic symptoms, the fever, the chills.
Right, the rigors, all correlated with the synchronized bursting of red blood cells.
But the deadliest species, Plasmodium falciparum, has an additional lethal trick up its sleeve, responsible for severe complications like cerebral malaria.
P. falciparum is unique because it alters the surface of the infected RBC. It causes the host cell to display specialized adhesion proteins, like ITM1 and CD36. These adhesion proteins facilitate cytoadherence.
The sticking of infected RBCs to the endothelial lining of blood vessels.
Exactly.
Why does the parasite want the infected RBCs to stick?
For two critical reasons. First, to evade the host's primary clearance mechanism, the spleen, which constantly filters and destroys abnormal or infected RBCs. If they stick to the vessel walls, they avoid splenic filtration.
They hide from the filter.
Right. And second, when these infected RBCs stick in the small capillaries of the brain, they cause vascular obstruction. This leads to the ischemia, coma, and multi-organ damage characteristic of cerebral malaria. The pathogen is actively manipulating the host cell to facilitate its survival and cause profound damage.
This evolutionary pressure is so strong that human genetics has actually evolved counter-defenses.
Precisely. We see natural selection favoring host resistance traits, such as Duffy blood group negativity, which prevents P. vivax entry into RBCs, and sickle cell hemoglobin, HBS. Though HBS causes the disease itself, it confers protection against severe P. falciparum malaria.
Which reflects the immense selective pressure this parasite imposes.
It really does.
Next, toxoplasmosis, Toxoplasma gondii, famous for its association with cats, the definitive host.
Toxoplasma has a complex cycle. Humans and other animals are intermediate hosts, typically acquiring the parasite by ingesting oocysts from cat feces or tissue cysts from undercooked meat. While generally asymptomatic in healthy individuals, it holds severe risk for two key populations.
Immunocompromised patients, where the latent infection can reactivate.
Yes, in patients with AIDS or transplants, the dormant parasite forms, we call them bradyzotes, stored in tissue cysts in muscle, heart, or brain can reactivate, leading to severe encephalitis.
And the other population?
Pregnant women. A primary infection can lead to devastating congenital toxoplasmosis, causing hydrocephalus, cerebral calcifications, and severe developmental issues in the newborn. The ability of the organism to form these lifelong tissue cysts is the core of its long-term pathogenesis.
Our final parasitic example is schistosomiasis, Schistosoma spp, a disease that affects 230 million people globally, where the pathology is almost entirely driven by the host's reaction, not the worm itself.
This is a classic example of severe immunopathology. The adult worms live in the portal or bladder veins, but the clinical disease is caused by the massive number of eggs they secrete. These eggs often become trapped in host tissues, the liver or the bladder wall, instead of being excreted.
So the worms are living there, but it's the foreign material, the eggs, that causes the destruction.
Correct. The trapped eggs provoke a chronic, intense, granulomatous inflammatory reaction, a dense walling off process similar to what we saw with TB. If the eggs lodge in the liver, the chronic inflammation and granuloma formation lead to pipe stem fibrosis and portal hypertension.
And if they lodge in the bladder?
Chronic inflammation and hematuria result. The damage is a direct result of the host's prolonged and ineffective attempt to eliminate the durable foreign egg material.
Okay, the battlefield is never static. New threats emerge constantly, challenging our public health systems. This brings us to the modern context of infectious diseases and the sophisticated technologies we now use to identify these complex invaders.
Absolutely. Understanding pathogenesis means understanding evolution. The emergence of new pathogens is intrinsically linked to global change.
The critical factors driving the rise of emerging infectious diseases, or EIDs, are pretty clear. Environmental changes altering vector habitats, massive human demographic shifts like population growth and rapid urbanization.
Breakdowns in public health infrastructure, and critically, the speed of global travel and movement of people and animals.
These factors create opportunities for zoonotic spillover pathogens crossing from animal reservoirs into humans and for subsequent rapid global dissemination.
We have witnessed the cycle repeatedly since the 1970s.
Looking at the historical context, we see critical examples. Ebola, first identified in 1977, and terrifyingly re-emergent in 2014. HIV appearing in 1983, SARS in 2003, Zika in 2007, and most recently, the SARS-CoV-2 virus causing COVID-19 in 2019. Each case demonstrates how new ecological niches or human behaviors fuel disease emergence.
And this leads us to the reality of bioterrorism agents. Public health agencies must classify these agents based on their potential for mortality, ease of transmission, and the level of public health preparedness required.
Category A agents represent the highest risk. These can be easily disseminated or transmitted person to person, resulting in high mortality and requiring significant public health action.
Right, and this category includes historical scourges like anthrax, smallpox, and plague. Understanding the pathogenesis of these agents is crucial not just for natural outbreaks, but for defense against deliberate threats.
Given the speed and complexity of these threats, rapid and accurate identification of the pathogen is vital for initiating effective treatment. What's the current hierarchy of diagnostic techniques?
Traditional methods remain foundational, but modern molecular tools have greatly enhanced speed and precision. We begin with microscopy and staining.
These initial steps give instant clues about the microbe's classification and morphology.
Absolutely. The Gram stain immediately classifies most bacteria based on their cell wall structure, but for specialized pathogens, we need specialized stains. The acid-fast stain is essential for visualizing the unique waxy cell walls of mycobacterium species, like TB.
And for parasites?
For visualizing parasites and inclusion bodies within host cells, we often rely on the GEMSA stain. It's critical for identifying leishmania, malaria, and inclusion bodies related to chlamydia or certain viruses. And while often slow, culture remains essential for sensitivity testing and isolating the live organism.
But the real diagnostic revolution has come through molecular techniques which bypass the slow growth of culture.
The cornerstone of rapid modern diagnostics is nucleic acid amplification, primarily the polymerase chain reaction, or PCR. PCR allows us to take tiny amounts of microbial DNA or RNA from a patient sample and amplify it exponentially, allowing for detection even when the pathogen load is very low.
When is PCR most critical?
It's vital when speed is paramount, such as diagnosing herpes simplex virus encephalitis in the brain, or for identifying fastidious organisms that are difficult or slow to culture, like chlamydia. PCR gives us a definitive answer in hours, not days or weeks.
And looking ahead, the source and highlight next-generation sequencing, or NGS, as the emerging technology. How is NGS different from PCR?
PCR requires us to know what we are looking for. We have to design primers specific to a known sequence. NGS, by contrast, is a massive parallel sequencing technology that allows us to sequence virtually all DNA or RNA present in a patient sample simultaneously.
So you don't need a suspect. You just sequence everything.
Exactly. This means we can rapidly identify not only known pathogens, but also completely novel or emerging ones without needing prior suspicion. While currently more specialized, NGS holds immense promise for comprehensive, unbiased pathogen identification, significantly changing how we characterize outbreaks in the future.
So what does this all mean?
Yeah, where do we land?
We began this deep dive seeking the rules of engagement, and we've uncovered a dynamic, complex evolutionary war. The success of a microbe depends on its ability to overcome epithelial barriers, deploy virulence factors like powerful toxins or sophisticated antigenic variation, and manage complex life cycles.
And the key realization, I think, is that disease, in its most severe forms, is frequently an equation involving two factors: the direct damage of the microbe and the catastrophic collateral damage of the host's response.
Right. Systemic shock from sepsis, the progressive tissue destruction in chronic viral hepatitis, or the cavitation in secondary TB.
These are often manifestations of the body trying desperately but destructively to contain an enemy it cannot quickly defeat.
And as we close this deep dive, let's return to the chronic diseases we discussed, tuberculosis and syphilis, which can linger for decades. These pathogens, M. tuberculosis and T. pallidum, they don't rely on the fast, powerful, immediate damage of superantigens.
No, they don't.
Instead, their virulence is defined by their capacity to induce a specific, prolonged T-cell-mediated response, the granuloma or the gamma, that ultimately causes slow but extensive tissue damage.
So this raises the critical question for you to mull over. In the context of persistent chronic infections, is the most successful pathogen the one that simply possesses the biggest, fastest weapon?
Or the one that has mastered the art of making the host hurt itself?